It is my absolute pleasure to invite up to the podium Dr. Sumanth Prabhu. He's the Lewin Distinguished Professor of Cardiovascular Diseases, Professor of Medicine, Pathology and Immunology, and Chief of Cardiology at Washington University in St. Louis. A heart failure cardiologist and physician scientist, his research has been central to advancing the field of cardioimmunology, focusing on how immune and inflammatory mechanisms drive heart failure. His lab has defined the role of a cardiosplenic axis after myocardial infarction and uncovered key immune cell drivers of para-inflammatory responses in heart failure. Dr. Prabhu is an elected member of several leading medical societies and currently serves as Senior Associate Editor of Circulation Research. Please welcome.
Well, thank you very much. Thanks again, Slava, for the invitation to present. It's a real pleasure to be at this conference, seeing such nice science being showcased, and a real honor for me to talk about some of our work. My work focuses on heart failure, and this should be playing as a video. I'm not sure. There we go. Well, the normal heart's not playing, but I think you can see the failing heart is quite dysfunctional. And if you look closely, this is an ischemic cardiomyopathy with apical akinesis. And the process that leads from a normal heart to a failing heart, we term cardiac remodeling. And we've known for a number of years that there's a strong inflammatory component to this process. And indeed, if you look at what is happening at the tissue level in this failing heart, what we see is that there is a uniform expansion of both the cellular and non-cellular effectors of the innate immune system, as well as the adaptive immune system. And within failing myocardium, there are populations of biologically active and tissue destructive innate immune cells, such as macrophages, and adaptive immune cells such as T cells. We had proposed that this was the result of a so-called para-inflammatory response, modeling it after the concept proposed by Medzitov in a classic paper in the earlier 2000s, that the heart, after following an acute injury where there's intense inflammation, if the infarct in this case is large enough to induce secondary responses such as neurohormonal activation or mechanical overload, instead of complete resolution of inflammation, there's ongoing low levels of chronic injury and inflammatory responses that are termed para-inflammation that results then in expansion of resident cells. So para-inflammation is a low-grade immune response that occurs when the tissue is under stress. And so we were interested as to what constitutes the para-inflammatory response at the tissue level in the failing heart. And work done by Slava, Corey Levine, Doug Mann, and others. I'm sure many of you are aware of the classification of macrophages in the heart, that they can be divided into three groups based on their expression of a chemokine receptor CCR2 and MHC2 into three populations, including a CCR2 negative population, which could be MHC2 low or high, and a CCR2 positive population, generally MHC2 high. The CCR2 negative positive population is derived from monocyte infiltrating monocytes and monocyte-derived macrophages. And the MHC2 low CCR2 negative population is derived from embryonic cells, embryonic macrophages. While the third category can be derived from either group, depending on the pathophysiological context. So there was a study done by Nerendorf's group that if you look in chronic heart failure, now this is not after acute myocardial infarction, but in the chronic ischemic cardiomyopathic state, unfortunately I don't have a pointer, but they used a dual reporter model whereby they could separate out locally sourced versus infiltrating macrophages based on fluorescence of YFP and I believe RFP in this model. It's hard for me to see the slide. But if they looked at a chronically failing heart, there was an increase in the monocyte-derived population, a significant increase, but most of the cells were still locally sourced in the myocardium. And a simplistic way to look at it is just CCR2 negative and positive cells. And you can see that in heart failure, this is from a mouse model 8 weeks after myocardial infarction, in heart failure versus a sham-operated mouse, if you look in the heart, there's an increase in CCR2 positive cells and a reduction in overall CCR2 negative cells, but the CCR2 negative cells are still the predominant macrophage population. So single-cell analyses have shown that resident macrophages express a variety of markers, including TIMD4 and LIVE1 and 4 receptor 2, but also express markers that are classically in the past have been, quote, M2-like markers such as CD206 and CD163. And in the status of ischemic cardiomyopathy, these macrophages have classically been alternatively activated macrophages, activated by Th2 cytokines. And we wanted to examine whether alternative activation of macrophages plays a role in the tissue remodeling that we saw in the first slide. So alternative macrophage activation occurs via Th2 cytokines, such as IL-4, IL-13, and IL-10, and downstream signaling that activates and transcribes genes classically associated with M2 macrophages in vitro. And this was important to us because we had previously shown in the study of adaptive immunity in the failing heart that if we look at the T cell populations that are expanded in failing myocardium, there's a predominance of Th2 versus Th1 CD4 positive lymphocytes. So we wondered if there was downstream or rather cell-to-cell effects of Th2 cytokines on macrophages. So the hypothesis here was that chronic Th2 alternative activation of CD206 macrophages in the failing heart induces this para-inflammatory response in LV remodeling. So it's just 3 points I'd like to make. And the first one is that cardiac macrophages in the post-MI heart is a alternatively activated para-inflammatory immunofibrotic response. So here we took mice that underwent coronary ligation to induce ischemic cardiomyopathy or sham operation, and we simply quantitated the macrophage population at various time points after MI. And we used classical markers, including F480 and MERTK. You can see the representative flow plots there, and quantitation of total macrophages, and CD206 and CD positive and negative macrophages. And what you can see in that graph on the top left at the bottom of the slide is that total macrophages in a chronically failing heart, 8 weeks post myocardial infarction, the abundance of these cells is the same as 48 hours after myocardial infarction. So this is a very chronically inflamed and dynamically activated heart. And if we look at the macrophage subpopulations, now again using a classification of CD206 positive and negative, we see that the CD206 population there at the bottom left, it shows a progressive expansion over time, whereas the CD206 negative population is most abundant acutely after myocardial infarction and then drops to lower levels, and you can see the summative data there on the bottom right. Now, of the two receptors, IL-4, IL-13, all signal through IL-4 receptor alpha, and IL-10 signals through IL-10 receptor alpha. What you see here is that of the alternatively activated cytokine receptors, the predominance, about 40 to 50% of CD206 macrophages, express IL-4 receptor alpha progressively, the expression progressively increases throughout the development of ischemic cardiomyopathy. So to characterize these cells a little bit better, we looked at markers that LIVE1 and MHC2. We also looked at the proliferation rates using Ki67. And we found that, first off, that most of these cells were LIVE1 positive and MHC2 positive and CCR2 negative, although the proportion of CCR2 negative cells increased in chronic heart failure. And so they would represent that middle population of that classification. And although we didn't lineage trace these cells, we feel that they're derived from both resident cells and monocyte-derived cells. And indeed, studies have shown, including Slava's paper in Nature Immunology, that monocyte-derived cells assume resident-like fates with chronic disease in ischemic cardiomyopathy. So we then looked at the spatial distribution of these cells, and I apologize I don't have a pointer here, but the red immunofluorescence represents CD206, and the green represents collagen hybridizing peptide, which marks areas of collagen turnover and collagen deposition. And what you see here is that broken up into the infarct, border, and remote zones, that almost the majority of these macrophages are always associated with areas of collagen turnover. So there's a close link between CD206 macrophages and fibrosis in the heart, especially in the infarct and border zone, and also in the remote zone, although to a lesser extent. Again, we use Ki-67 to assess proliferation, and interestingly, only the CD206 population was Ki-67 positive. In other words, this was the exclusively proliferative macrophage population. So the lack of expression of CD206, there was no macrophage proliferation noted, at least using Ki-67 as a marker. And spatially, the proliferation of these macrophages, again, broken down by infarct, border, and remote zone, primarily occurred in the infarct and border zones, and the proliferation rates in the remote zone was equivalent to sham-operated hearts. So this suggests that these macrophages are dynamically expanding, associated with fibrotic regions, and may be responsible for the infarct expansion and post-infarct remodeling that we see very frequently in animal models and in patients. So we felt that this was evidence of a Th2M2-like immunofibrotic response in the failing heart. So the next question was, what is it about these macrophages that is contributing to a fibrotic response? And here we took bone marrow-derived macrophages, polarized them with either nothing, which would be M0, or IL-4 or IL-10. And you can see by flow, all these cells are essentially CD206 positive. And this is just macrophage gene expression 24 hours after cytokine polarization. And what you'll see here is that we have expression of classical M2 markers, but that IL-4 polarization led to a selective, very, very extraordinary upregulation of this protein, of this gene called FISI1, which is part of the resistant pathway, or resistant-like molecule alpha. And we'll talk about this a little bit later. But if we did a collagen gel contraction assay, these collagen gels are imbued with mesenchymal stem cells here. We've also done it with cardiac fibroblasts, so that if there's a myofibroblastic phenotypic switch, there's a contractile phenotype and the collagen gels will contract, and this is an assay for myofibroblast differentiation. What you see here is that if you expose these collagen gels imbued with mesenchymal stem cells to conditioned media from these macrophage polarized groups, it is only the IL-4 polarized macrophages that induce a strong myofibroblast differentiation response. And this was shown by both the physiologic assay as well as smooth muscle actin expression. So we know then that these cells are expanding, they're exclusively proliferative, they're associated with fibrosis, and they secrete very high levels of FISI1, and they induce myofibroblast differentiation. So the second point is that these macrophages are sufficient to induce LV remodeling and fibrosis. So here we took those same bone marrow-derived polarized macrophages, used isotype mismatched mice so we could track donor cells, and we did adoptive transfer with intramyocardial injection into naive recipients. So we waited then 4 weeks, and here's an M-mode echocardiogram, and what I hope you could see is that of all the groups, the adoptive transfer of macrophages polarized by IL-4 led to LV dilatation and LV systolic dysfunction over a 4-week period. This was progressive and evident only in this group compared to the others. And these mice also showed evidence of left ventricular hypertrophy with increased LV mass. And if we looked at the tissue level, we saw a similar fibrotic response that was accentuated in the IL-4 transferred macrophage hearts. And it's quantitated there, but there was a clear evidence by histology. And if we use the same assay with collagen hybridizing peptide, we found that these macrophages were associated with zones of collagen deposition and turnover, and incredibly, this was occurring with both donor-derived macrophages And this is 4 weeks later, so there's persistence of these macrophages, as well as recipient-derived macrophages. So these macrophages are presenting a microenvironment that includes expansion of recipient resident cells. Looking just a little further, I apologize, I don't have a pointer, but we looked at myocyte hypertrophy, myocyte apoptosis, and then the abundance of these transferred macrophages based on recipient and donor origin. And so these cell transfer resulted in myocyte hypertrophy, resulted in increased cardiac cell death, and expansion of CD206 macrophages. Now, this expansion occurred in all, the persistence of donor cells occurred in all of the adoptively transferred groups. But only the IL-4 polarized macrophages led to an expansion of recipient macrophages, again, indicating that this was a microenvironment that we could induce in the naive recipients to produce features of LV remodeling. So the third point, sorry it's mislabeled, is that we could then use this and leverage it to have IL-4 receptor-directed therapeutics in heart failure. And so just a step back, if we do a flow cytometry of all cardiac leukocytes that express IL-4 receptor alpha, and then assess how many of those cells are macrophages, we see that the majority, over 90% of IL-4 receptor alpha cells, expressing cells in the heart, are macrophages. And they are primarily CD206 positive macrophages. And we also looked at multiple other immune cell types, including neutrophils, Ly6E high monocytes, Ly6E low monocytes, not shown here, but CD4 T cells. And they don't express very high levels of IL-4 receptor alpha. The most was in Ly6E high monocytes, about 5%. So if we use therapeutics targeted against IL-4 receptor alpha, it's fairly specific for the expanded macrophage population in the failing heart. So here we used antisense technology, and we used antisense oligonucleotides directed against IL-4 receptor alpha and administered it in a mouse model with chronic ischemic cardiomyopathy. This is 4 weeks after myocardial infarctions. This is heart failure, not after acute MI. And we delivered this on a every 3-day basis and followed the animals weekly. And these are 2 echocardiograms showing a mismatched oligo control delivery. And what you could see, I hope, in the echocardiograms is a persistence and progression of LV dilatation and dysfunction in these infarcted hearts. And then take a look at the ASO, IL-4 receptor alpha ASO hearts. What I hope you see is that there's as much of an infarction to start out with, but that the size and the function of these hearts improve over a 4-week period, indicating reverse remodeling. And that is quantitated here. Some reason the gross pathology is not showing up there, but we saw a decrease in the heart weights and lung weights. We saw a progressive improvement in chamber volumes and a progressive improvement in LV ejection fraction with IL-4 receptor alpha ASO treatment, and a reduction, incredibly, of fibrosis, especially in the border zone. And we quantified myocyte hypertrophy using histology as well. Essentially, all of the aspects of tissue remodeling as well as chamber function were reversed with this therapy. Now, this silencing also depleted the cardiac macrophage population in the heart, both the total macrophage population and the CD206 macrophage population and the IL-4 receptor alpha expressing population in the heart. And it reduced macrophage proliferation. So blocking this receptor reduced the expansion of macrophages with the progression of ischemic cardiomyopathy. And it did so without a change in the ligand for these receptors. And IL-4 serum levels were unchanged. IL-13 was not even significantly elevated. But pro-inflammatory cytokines, so-called Th1 cytokines, and chemokines were reduced across the board, suggesting a systemic anti-inflammatory response with IL-4 receptor alpha-directed therapeutics. Just going back to PhysiOne, here we did a histological analysis of the treated groups for CD206, IL-4 positive, PhysiOne positive macrophages with these oligos. And the long upshot is that giving this therapeutic reduced the number of CD206 positive cells, CD206 positive IL-4 positive cells, and CD206 positive IL-4 positive FISI-1 positive cells. And almost all of the IL-4 receptor expressing cells were FISI-1 positive. So we went back to our collagen gel contracting assay and redid it with MIL-4 polarized conditioned medium, but here we blocked FISI-1 with two approaches, one with an anti-FISI-1 and the other using FISI-1 knockout bone marrow-derived macrophages. And we found that the myofibroblastic activation was abrogated if we blocked this secreted protein that was so markedly upregulated by IL-4 polarization. So the conclusion is that then after MI and ischemic cardiomyopathy, CD206 positive macrophages are proliferative, progressively expand, and they comprise the predominant macrophage population of the failing heart, and they closely associated with areas of fibrosis. They're primarily CCR2 negative, LIVE1 positive, with a sizable population expressing IL-4 receptor alpha. There's also increased levels of IL-4 in heart failure. And this polarization with IL-4 is sufficient to induce upregulation of FISI-1 and FISI-1-dependent myofibroblast differentiation. And these macrophages are both necessary and sufficient for the pathogenesis of adverse remodeling. And we can leverage this therapeutically by silencing expression of the IL-4 receptor alpha. It is primarily targeted towards failing heart macrophages expressing CD206 and induces a reversal of LV remodeling. So the implications then is that targeting an alternative activated cytokine pathway, IL-4, IL-4 receptor alpha, in these CD206 macrophages might be a new anti-inflammatory immunomodulatory approach to limit cardiac remodeling in heart failure. So this is a different perspective than the traditional Th1 cytokine antagonism that has been so widely proposed. So let's stop there. I'd like to thank my laboratory, including and especially Shangsheng Wang, who drove most of these studies. She's now assistant professor at Wuhan University in China, the lab members I have, and the sources of funding. I appreciate the invitation. Happy to take questions. Thank you.